So welcome, Tom and Dmitri. It's a
Thank you.
pleasure to have some time to chat with you. This is part 2 for Tom, but we're just getting started here with Dmitri. So Dmitri, if you could just give us an introduction, tell us a little bit about yourself, your background, your training, your current position, and then maybe a little bit about your research interests.
Okay. Well, first off, it's great to meet you. I've read some of your papers, and they were the foundation for our research. And the system that we're using in our research is the IBM system, both the X10 and X24. I am trained as an experimental psychologist. I'm actually the graduate of the University of North Dakota. For 7 years, up to 2003, I worked for a medical school as a research analyst. And then I got hired back at the psychology department of the University of North Dakota in a teaching/research position, faculty position. And I've been primarily teaching, well, things like perception, physiology of behavior and psychophysiological measurements, biological basis of behavior, and multivariate statistics. My research interests are, well, have been shaped by going to too many hockey games and hanging out with an optometrist who suggested that we should start testing those hockey players with some physiological tools, looking at some oculomotor functioning of their visual system, and see how that corresponds to their performance. So we started doing that, and then incorporating more and more toys into our assembly of tools that we use. Pretty soon we started looking at EEG as well as visually evoked potentials, VEPs. And we got funded by the North Dakota Department of Commerce, the so-called Research ND program, where we looked at youth athletes, youth hockey players, those with a history of concussion and those without a history of concussion. That was a big study. So we baseline assessed them on a number of instruments, including what they call the Nike Sensory Station, now known as the Synaptic Sensory Station. It's by the same Nike group, but apparently after 2015 there was some internal issues. They closed down the program. But it's a kind of like a psychomotor type station that assesses multiple visuomotor skills. And our first baseline assessment, we used ABM's system to look at perhaps baseline differences between those with a history of concussion and those without the history of concussion, simply by looking at their theta to gamma ratios at the FZ site during visuomotor tasks that included things like go, no-go, perception, span, hand-eye coordination, all that. So there is a kind of a working memory with a temporal constraint component. And the reason we were using that ratio was because of previous studies when they tried to time lock gamma cycles within the theta cycle, and they were able to predict even working memory span. And we thought that in the absence of perhaps behavioral differences on reaction time and accuracy indices of their performance, we could find some type of EEG deficits. And sure enough, we did. And we published in scientific reports that they had lower theta to gamma ratios, suggesting working memory deficits.
Yes.
Yeah.
And so just to clarify, when you had a measurement of working memory, a behavioral measurement, along with the EEG measure.
Yeah, simultaneous.
The EEG measure was more sensitive.
Right.
Was it more sensitive to the concussion effects than just the pure behavior measure?
Yeah, to the history of concussion. They were all at least 6 months after their most recent concussion. Yes.
Right. Okay.
We found a similar thing with VEPs as well. We looked at their P100 wave and looked at it in the environment of two different types of stimuli. One type of stimulus was kind of like stimulating their parvocellular tract, and that's a high contrast checkerboard pattern that is reversing at a very low rate. The other type of stimulus was stimulating their magnocellular tract,
Right.
using very low contrast, like 10% contrast sinusoidal gradients that would reverse at a high rate. What we found with their VEPs is that their P100 was both the amplitude and the latency was significantly smaller.
For both of those stimulus sets, you saw latency and amplitude.
Yeah, suggesting that they have some persistent visual deficits in magnocellular processing that has to do with motion detection and anything that's relevant to motion processing. So they were not as efficient. Right.
And was this also, what was the window post concussion for that?
Yeah, also six months or so.
Okay. And then did you follow those participants?
Good question. So we actually trained them using two different protocols of what we call standard optometric vision therapy that targets oculomotor training of the oculomotor system, the so-called hardware, your saccadic eye movements, your vergences, as well as accommodation. And then we followed it up with what they typically use in sports vision training. That's more kind of like the intersection of your visual processing with attention, with decision making, all of those things. And so for one group, all of our players completed, I think it was 8 weeks of training. No, actually 10, considering that we also tested them for 1 week. Yeah, 10 weeks of training in both type of programs, but we reversed the order for half of them. So to see if there was any order effect as well. And also collected their VEP data, their Eeg data throughout at the point when we reversed the training order. So we switched them 4 weeks and 4 weeks, like the blocks, or if you want to consider 5 weeks and 5 weeks, and tested them again. And that is where I am right now analyzing the data. I've just finished analyzing the VEP data, and it's looking good, just like we suggested. In fact, one type of program was more beneficial for retraining of the magnocellular pathway than the other.
Meaning you started to see some restoration of the P100 and the latency wasn't as late.
Right, exactly.
Did you also look again at the working memory and the
that's my next series of analyses. So I'll be looking at that as well, yes.
Interesting.
But this is not just the only study that we did with your system. My graduate student who is now working for Boeing, he just graduated, and he's a human factors engineer now. But yeah, he was looking at air traffic controllers. Kyle, yeah, you know Kyle, yes.
Yeah, great guy.
Yeah, he is brilliant. He helped us out quite a bit in processing all our data and analyzing the data. So
he was also working on the concussion study, or he had a completely different study as well?
Yes, he had his own where I participated in an advisory second author type capacity. So, but he was looking at air traffic controllers as well as at kind of like a simulation of the MAD-B, that application, the NASA MAD-B battery. And he was looking at workload, really. That's his thing, and engagement. And what we did find in one of our studies using MAD-B that links his type of research to mine was that we used a measure called Convergence Insufficiency Symptom Survey. It's like a 15 item questionnaire that is a really good screener for oculomotor issues, like accommodative insufficiency or convergence insufficiency that you oftentimes see not just in, say, kind of ADHD population. There is a huge comorbidity of oculomotor disorders and ADHD, but you also see it as a persistent deficit of concussion, that the oculomotor system becomes inefficient in eye team and invergences, in eye movements, etc. And what we found in that study, when we equipped them with EEG during completion of the MAD-B task, half of their task was automated, half of their task they had to do some basic monitoring of the system. And so by the time of the automated component, it's like a 40-minute task. The last 10 minutes, we analyzed it to see if they're going to develop kind of like boredom and start to idle, so their engagement goes down.
Right.
We didn't analyze ABM's indices of workload and engagement, but we simply used the alpha at POZ, and it was diminished for the automated condition in the last 2 minutes of the task. So, which is kind of interesting, because we're thinking now of using TDCS, transcranial direct current stimulation, to see if we can modulate those things with different type of stimulation protocols.
Right. Have you done work with TDCS?
Yeah, a little bit. Just in kind of like a pilot studies. I did a workshop at Baron. The Harvard Med School Center for transcranial magnetic stimulation. Yeah, I did a workshop there on brain stimulation, and have tried to incorporate it into some of my studies.
Interesting.
Finally, the other line of research, since I'm kind of like the psychophys guy in our department, when professors wants to add another line of evidence to their behavioral cognitive protocols, they would ask me to help out.
To you.
Right. So one study that we did with the elderly, actually, we were looking at positivity bias in memory, more towards recall of positive memories as opposed to negative ones in old and very old folks. And we also looked at their working memory and used a ABM system, and actually used your guys' indices of workload and engagement, and to compare the groups and the tasks. So it was a between group and within task type of comparison. So what we found in that study, that while the groups did not really differ much in terms of the memory, in terms of the workload was quite predictable from the demands of the task, and was nicely reflected in your output. So cognitive load and engagement, like the highest engagement was on a task where we would present them with emotional stimuli, so a bunch of pictures, and the lowest was when they were having to do some type of mental arithmetic type tasks.
And this was for age 65 and up, or older?
I think one group was up to 75, and the other one was like 65 to 75. One was 75 up. Yeah. So, and then the one that we did with Kyle and air traffic controllers, they had 4 distinct, and it was a kind of like a simulation protocol that they use in the aviation department. And so you have distinct phases of workload, really, when it's really easy, medium, high. And in addition to ABM's indices of workload and engagement, we also used pupillometry, eye tracking, and we measured pupil size. And there was a really nice correlation with pupil size and
and workload. Yeah.
Workload. Yep.
Yes, we've seen that as well. Yeah, we've been doing a lot of work with pupillometry recently. I think the eye trackers have gotten much better over the years. You know, we've been using the Tobii and
Yep.
finding really good results. We've been looking at social media posts and online videos and, you know, looking at the types of responses that get people's attention and keep their attention. And as you can imagine, it's, you know, the more salacious or evocative material, but we get a nice correlation between some of the EEG measures as well as the pupillometry that, you know, suggests just how engaging and arousing, I guess, you know, the different stimuli are. But it's nice to use the combined metrics for sure.
Yeah, yeah. So, and I mean, your guys' metrics have been useful as well. Again, I think the more studies we run, the more validated they become. I really think that,
yeah, I mean, I think your concussion work is really important and very relevant to the discussions that we're having today about athletes, whether they're, you know, professional athletes down to, you know, little leagues, and, you know, should I allow my child to play sports and at what age, and how can we assess what's happening to the brain when there is a concussion. And I don't think we have, as of yet, great biomarkers that suggest, okay, now is a good time, you can go back on the field, you're, you know, back to normal. So I think anything you can do in that domain will be greatly appreciated, you know, by the sports community and, you know, all the concerned parents. It's tricky research because you have to follow, you know, from the concussion, you know, for as long as you know, is feasible. And those kinds of studies always take a lot of time and are expensive.
Yeah, absolutely. Yeah.
But it's interesting that you're doing some interventions as well. So when you did the oculomotor interventions, how frequently did they come into your lab to do it? Or is it something they could do at home?
Fortunately, my wife is a vision therapist and sports vision trainer.
Oh, okay. Well, that's a good collaboration, huh?
Yeah, so we put her on a grant and she did it for us.
So these are interventions that she does routinely.
Yeah, we just created a protocol for her. That's based nice. On what she does. And our supervising optometrist is my research collaborator with whom, you know, we started doing all these type of studies. So it's a very good collaboration, since I'm kind of like the science guy and they're all on the clinical side, and they have all the equipment as well. It's not like we have all kinds of toys here. They have all those type of stations and gadgets that oftentimes are just sitting around and not being used.
Yes. Oh, yes, definitely. Does your wife perceive the need for an objective measurement of these? I mean, she probably knows about the outcome because she's worked with them a lot, but if she had a relatively simple EEG measure to document the outcome, would that be useful for her?
Yeah. So the VEP system that they're using is the diopsis. By diopsis, I believe they're the largest one. And they have multiple modules. A lot of them have been kind of FDA cleared for different type of things, like what do they do it for? Some just retinal pathologies that they use. But so I previously worked with another optometrist who created those magno and parvo stimuli, and he pitched it to diopsis, and they created for us an experimental module, essentially. And that's the module we've been using. And now she is actually my wife is using it in pre and post assessments. And even insurance companies are starting to recognize those VEP assessments as valid. But a couple of issues from looking at, and you probably know that as well, that when you look at those VEP data, especially when it comes to amplitudes, you see a lot of noise. There's a lot of variance there.
Sure. Sure.
Latency is probably a better kind of a metric to go by.
Mm-hmm. But generally within an individual, of course, then, you know, ideally in an individual, you'd have a baseline established measure before they ever hit their head. And then you would be able to track them, because generally, you know, there's a pretty good fingerprint both for the event-related potentials as well as the power spectral densities for each individual. But you need to be able to assess them in the intact state, and that's not always possible.
Yeah, which in a clinical setting is not an option, because a lot of them, they're like WSIs, work comp type
Sure.
referrals,
Sure.
you know, from car accidents and what have you. So there is no baseline. But, you know, Dave, my optometric partner, has started actually routinely just running all his patients on VEP, just in case, you know, they have
That's great.
Yeah,
That's great.
Yeah.
That's great.
But definitely it's not the best, most accurate marker right now. And so, yeah, there's potentially something
But really,
else.
there are no established useful blood or other biomarkers, and you can certainly go through both CT and MRI and not see what you might see with the EEG, because it's assessing, you know, the way the neurons are communicating, you know, as opposed to a gross structural damage. So we're keenly interested in that area. We've been asked at various times. We did a little bit of work with the NFL and a couple other sports teams. You know, is there something you could give us that we could have on the field as an assessment? I don't think we're there yet, but I do think it's feasible, you know, for the future. So it's really exciting to hear about your work in that area, and hopefully you'll continue to develop that. One question I like to ask everyone is, you know, if you suddenly found yourself the beneficiary of a very large amount of research funding, what are some of the dream research projects that you'd really like to accomplish?
Tom?
Oh, I already answered. This is your, I can't wait to hear your answer, buddy. So,
well, so I really think we're onto something when it comes to magnocellular processing and different type of pathology. I don't think magnocellular processing deficits are limited to concussion. What we also see now is that it may also be underlying the etiology of dyslexia. And the way you should approach dyslexia treatment and the standard for it oftentimes is phonemics based, and really has to do with grapheme phoneme type correspondence training, whereas more and more studies show that they may suffer from similar, a lot of dyslexic may suffer from similar magnocellular processing deficits and have very inefficient saccadic movements and inefficiently process that motion, eye movement related type information. So we just got another grant from Research ND that, and Tom is on it.
That's great.
Where we're looking at basically comparing the two types of treatments, the standard, you know, the gold standard of care treatment for dyslexia with magnocellular training, and we're going to be collecting, you know, EEG stuff.
That's fantastic.
Yeah. So with them as well. So, but if I had more funding, we have an FNIR system. I want to see what kind of correlates we can find with you know, blood oxygenation flow and EEG information.
Do you have the whole head or the frontal?
Well, the cap is the whole head, but we only have so many optodes, so we have to kind of like configure them either depending upon what your area of interest is. But yeah, I mean, I wish we had. Even when we were rich, we were cheap. We couldn't afford the whole head. So, and I want to also combine it with TDCS and see how those type of modulations, how they affect EEG metrics, FNIRS.
Yeah, I think that the FNIRS is very interesting. We've been playing around with it ourselves and trying to put the EEG and the FNIRS on at the same time, which is doable. And certainly NIRS is going to give you a much better spatial localization, not as good of a temporal. You know, you don't pick up the fast millisecond level time changes like you do with EEG, but it does give you a much better spatial localization. The thing I think we're all struggling with, because there's a lot of research now on NIRS, is we don't have the library of artifacts and how to deal with artifacts that we do with EEG. I mean, EEG is 100 years old, right? So we've had a lot of time to figure out how do we decontaminate, and a lot less time on NIRS. So you still get, you know, if you have gross head movements or other muscle movements, you're still going to get contamination of the NIRS signal, and there's no easy ways yet to decontaminate. But I know there's a lot of great engineers out there working on it, and ultimately, to me, I'd like to have the EEG and the NIRS simultaneously, because it's kind of two sides of neural activity, basically.
Yeah. So that, exactly. So you get both the spatial and temporal resolution.
Right, exactly. Yeah. Tom, have you been working with NIRS? I don't know if we talked about it.
No, I have not. That's been Dmitri's gig. I haven't jumped into that at all.
We only use it in one study so far, so the NIR-X system, that's the one that we use.
Yes, that's the one we have too. Yeah.
Okay.
Yeah.
Well, their software is not very friendly. I mean, you can run like SPM1 and 2 analysis within the software on your subjects, but we had trouble kind of like exporting emergent data.
We've had our share of challenges too. But again, it's a relatively new technology, so you know, there's only a handful of manufacturers, and I don't think, you know, the software will come along. We need a lot of smart graduate students and research fellows who have lots of time to, you know, help them develop the software and the tools for analysis.
Yeah.
So what, like Tom and Dmitri, what are some of the projects that you collaborated on? Did you work together on the concussion study, I guess?
No, not the concussion. We worked together on the air traffic control.
Oh, the air traffic control, sure.
We worked together on where the ones I may have mentioned to you, where we're measuring EEG in flight and measuring EEG for learning predator training. We're working together on those. I hope now that I have that LabX software and I'm getting smarter by the day, if and when we ever get to run humans, I'm hoping to talk him into collaborating on more educational applications of your system.
Well, right now we're not running any subjects clinically with the dyslexia grant, but Tom is also.
Yeah, and we collaborate on that. I'm also in my spare time. I run a clinic where I evaluate dyslexia and ADHD and learning problems, and so I'm Dimitri's designated evaluator.
Yeah, Tom also juggles quite a bit.
Yeah, one of those things from the 60s and 70s that you too much on your plate and not smart enough to pick and choose.
So have you been looking at EEG biomarkers for ADHD?
No, not yet. Well, Dmitri, you may have, right?
No, when I was doing my ADHD research, when I was a grad student, I was actually looking at cognitive measures and nicotine patches. So
we hope to start doing some of that, but the literature we've looked at only looks at biomarkers that I've seen published during resting EEG. I haven't seen anyone, and I may not have looked more recently, look at EEG as the person does different tasks that invoke working memory.
I think they do, like especially on corner CPT and theta waves and beta waves.
Oh,
There's
okay,
been
okay.
quite a bit of research published on that. We used back when we were just trying to understand what the EEG was all about. A NeuroSky's one electrode, you know,
Yes.
EEG set, and we used it in one a study with corner CPT and ADHD-like symptoms. We would induce those symptoms by making subjects wear -2 diopter lenses, so it will be harder for them to focus, and that would take energy away from processing of the signal to actually deciphering the signal and getting it to the brain. And we looked at both, you know, that one electrode EEG frontal, as well as accommodative lag. We had a pretty sophisticated WEM 500 automatic refractor that would measure accommodative lag where they're focusing at, you know, millisecond by millisecond. What we found was that increase in error rate and other type of clinical indices